Hello, my name is Nathan Bosire Omari, and this is my presentation. We are looking into the fluorescence resonance energy transfer technique. It's also known as the FRET technique. So FRET, this is the introduction. So FRET is an imaging process that is distance dependent. Um, in this in this imaging technique, energy transfer happens non-radioactively from an excited fluorophore, which are also known as the donor, to another fluorophore, also known as the acceptor, through long-range intermolecular coupling. This long-range intermolecular coupling isn't really isn't known as long range due to the distance because it's as you can see the distance is very small. So FRET is a useful technique in showing protein interaction in living cells, and thus it enables the detection and monitoring of in vivo signal transduction and gene transcription. So for the fluorophores, a good example is the GFPs, which stands for green fluorescent proteins. So GFPs are common autofluorescence proteins used as donor and acceptor fluorophores. So this image over here in this page is an image of glutamate triggered long distance calcium signal transmission. We can see from left to right that as time increases, the figure tends to get more and more yellow. So this image shows an application of a glutamate to a cut surface of a leaf. So we can see that we can see from the data that the calcium concentration increases with time, and thus we can also see that the glutamate, and thus we can hypothesize that the glutamate triggers long distance calcium signal transmission. So in all FRET experiments, there needs to be a fluorophore. So in this experiment, the fluorophore is a GCAMP3. So it's also the biosensor used in fluorescence and high resolution. So something interesting for FRET is that the high contrast and time tracking gives useful data on how the plant internal environment changes due to glutamate. So this data was collected in a live plant, and we can see that as the time went by, the intracellular calcium concentration increased. Um, uh, a disadvantage of the FRET experiment is that it fails to show everything else that's happening in the cell. It's so specific, so it kind of gives us an illusion that only one thing is happening. Like in the cell, it gives you the in this data, it gives you the illusion that there's only calcium, but no, there are many other ions involved. And the question that I have is, uh, is FRET a candidate as a method for detection of lysosomal storage diseases in humans? Like, can we use it for this purpose? Thank you for listening to my presentation.